That's perfect. Okay, so welcome to my TED Talk. Well, the thing is that I will introduce myself briefly because there've been a small mistake made in the introduction. I didn't expect introduction. So I was born and raised in Kharkiv, and I came to Czechia to study Czech language in 2013. And in 2014, I applied to this faculty, which I got admitted to. And then I spent, I don't know, 2 years sitting in this very room. And then for 7 years, I've been studying this faculty. In the third year, it became obvious to me that I guess like patients are more safe if I'm not around them in the same room, and that I'm more interested in biology and academical science. So I decided to apply to the Faculty of Life Sciences in parallel. I did not finish this degree because then eventually I had to focus on actually doing science. So basically, as you perfectly know, there is not much time while you're doing medical studies. There is not much time to invest into doing something else on the side. I'm mentioning this because since it's a 3-minute job event, maybe some of you that are interested in science may take it as an advice. And obviously, I could only do internships, science-related internships, in summers. So this is what I did. In 2017, I joined a great lab of Weizmann in Weizmann Institute, which focused on studying the bacteria in tumors. So they are very famous because they discovered that different types of tumors are inhabited by different types of bacteria, and these bacteria can convey chemical resistance or chemotherapy resistance to different types of cancer. Then in 2018, I joined Omri Wurzel lab again for 2 months to study regenerating worms. So these are weird animals that basically you can cut them in as many pieces as you want, and each piece will grow the whole worm. So they are quite exciting biological model. And then eventually in 2019, I joined for an Erasmus stay. I joined my current lab where now I'm doing PhD. But before doing that, I actually had to stop my studies because I wanted to try to do science full time. And for this, I had to take an academical leave, and I've joined the lab of Nikolaus Rajewski in Berlin, which is where the Octopus project comes from. And since 2021, as has been said, I'm a PhD student in Barcelona. So now I would like in this lecture to talk about my current PhD project, which is in comb jellies, and the project that I did in octopuses. And you can very broadly assign these projects to 2 big questions that interest me personally, which is the evolution of cell types. So how did the cell types that we know in humans, where do they come from? And more importantly, where do the complex cell types come from, such as neurons? And the second question is, okay, once you have animals with different cell types, how comes that some animals become really smart? How do they build complex brains to become smart? And for this, we will talk about cephalopods. So before we go into that, I wanted to kind of defend myself, which is what people in my field have to do all the time, is basically explaining why do we have to study all different animals. And the most honest answer is curiosity, because people somehow assume that people are familiar with the idea that we have to study planets, we have to study galaxies, most of which we will never reach in our history. But then obviously we need to study the world around us, and studying other animals can also inform our bioconservation efforts. Second is that, okay, I get it, you may not be interested in anything that's non-human, but studying other animals gives us molecular tools. And one of the very important one in biology is green fluorescent protein, and this one got isolated from a jellyfish. This was a Nobel Prize. Another Nobel Prize tool is CRISPR. It's very popular now. It allows to edit genome in a very precise fashion, and CRISPR was isolated from bacteria. And we would never have gotten these tools if we weren't studying these organisms. Furthermore, studying different organisms can inform our understanding of human genome. So there has been a very intense progress over the last years in kind of building these neural networks that can predict protein function and structure and so on. And these models, they are trained on billions of protein sequences that obviously come and have to come from different organisms. So even when you sequence a genome of some weird butterfly, you actually indirectly informing the human research. So you know that the sequencing of human genome has started about 30 years ago, and this year it has been finished. I mean, I guess it's never finished, but this year they've published an update of human genome, filling all the gaps in the genome and so on. In terms of sequencing animals, there are also multiple efforts that aim at decoding the genomes of animals. So one of them that we in the lab really like is called Darwin Tree of Life, which is basically a British project where they aim to sequence all the eukaryotic species, about 70,000 of them, in UK. So all sponges, all trees, all mammals. And this is great that people realize the necessity to do this. One of the most interesting things, like one of the most interesting discoveries after we sequenced the human genome, after we gotten to sequence the genomes of different animals, was that animals are surprisingly genetically complex. And what I mean by that is that once the human genome was one of the first ones to be sequenced, humans turned out to have 20,000 genes. Well, it's kind of obvious that more primitive, quote-unquote, organisms, they have to have less genes. Well, this is absolutely not the case. So if you look at the sea anemone, for instance, which is undoubtedly more simple in terms of its body build and so on, than the mammals, like a mouse, we actually find out that they share about 80% of the genes, and they have roughly the same number of genes. And this is true for almost any animal. Roughly, if you sequence a genome, you will find 20,000 genes. So it's not the number of genes that makes us different. It's also not the identity of the genes, because again, they are mostly shared among these animals. What's important, in fact, is not the genome sequence itself. Yes, all features are encoded in the genome, but how this genetic information is interpreted by the molecular machines and how this is translated into the cell types. So once we are now passing this kind of age of just sequencing DNA, it's time to start making sense, also in a comparative way, meaning in other animals, not only humans, how this DNA translates to the difference in the body parts and anatomy that we observe in other organisms. And for this, we now have also we also have tools to study cells in a high throughput manner, and this is called single cell RNA sequencing. So the idea of the method is that we take a tissue that we are interested in, we dissociate into single cells, and then we profile every cell so that for each cell we see the expression of the genes in this cell. And this means that then if you profile thousands of different cells of different biological cell types, we can start grouping the cells into the cell types that are defined molecularly. So this data is usually summarized like this, where you take this multidimensional data of each cells and which genes they express and at which levels, and then you try to squish it into the 2 dimensions, where 2 dots in this plot, if they're close to each other, they have to have similar gene expression patterns. And basically, these blobs that you see here, they correspond to the different cell types. You can have muscles, you can have epithelial cells, you can have neurons, and so on. And so obviously, this was very fashionable. This has been very fashionable. So there has been this initiative established called Human Single Cell Atlas, the aim of which is to build the catalog of all cell types in human body, and there are a lot. So this is one of their mega plots, basically the concatenation of dozens or hundreds of studies, where you see these blobs corresponding to different organs from different donors and so on. So you see thyroid glands, cervix, esophagus, fetal organs, and so on. So there has been very intense progress in terms of mapping human cell types. Now they do it for different ethnic groups. There is a human cell atlas for Asia, human cell atlas for Africa. They do post-mortem samples. They do not newborn, but the fetal samples. But what about other species? And if you look at the amount of data sets, of the single cell data sets that have been generated over the last years, it grows exponentially. This is a logarithmic scale, right? It grows exponentially. And now some new studies, they can be, if studies, when I studied this faculty, a normal study contained 2,000 cells. Now it can contain well over the million. But if you look at, in terms of how we are, in terms of sequencing the cells from different species, you see that our efforts, the results are just pathetic. It's the over 90% of this data comes from human and mouse, not surprisingly because they're relevant for biomedicine. But then if you look at the non-vertebrate organisms, there's almost nothing, because these samples that come from non-vertebrates, they come from Drosophila and some more established model systems. So this is why in our lab, we kind of started this initiative, or want to start this initiative, which is called Biodiversity Cell Atlas, which is a parody on Human Cell Atlas, while if you just remove human and sequence everything that's not human, that you get a Biodiversity Cell Atlas. But the idea is to get from this extensive DNA sequencing to actually sequence the cells of different organisms in order to start making sense of how humans became humans, how it comes that we have hundreds of different cell types. So let's now look into one of the examples how people can study the origins of cell types. And for this, we will use animals that are called ctenophores. So if you look carefully, I don't know, maybe we should turn off the light, but maybe it's visible here, that they're transparent, and you see this kind of comb rows that are beaten. This is what gives them name. So ctenos means a comb, and these are just cilia, normal cilia that you find in many organisms and unicellular organisms as well. It's just that they're organized in these arrays and they're beaten synchronously. Why I'm mentioning this is because ctenophores are the biggest animals to move with cilia. We know unicellular organisms, but ctenophores can grow up as much like this, I'd say, and they still move with cilia. This is what makes them unique. Well, in general, if you look at them, you might think, oh yeah, that's like some weird jellyfish. You wouldn't be completely wrong. This is how people thought of them for centuries. So if you open any 19th century textbook and you go to the cnidarians, which are like corals, jellyfishes, and so on, the ones that are stingy and have tentacles, at the end you will find ctenophores, and it will usually say, yeah, ctenophores don't have stingy cells. They cannot sting you, but they have sticky cells. That's not much of a difference. They also have muscles, neurons, pretty much the same morphology we find in other cnidarians. Okay? Well, it came at a huge shock that when the first ctenophore genome got sequenced, that actually they're nothing like their supposed cnidarian brothers. They actually miss the majority of substantial proportion of neuronal genes. And even more, not is just they're missing some genes, is that these studies have placed them at the base of animal tree of life, which was very, very surprising. And why this is important? Because if we look at how the animal evolution was viewed before these studies, it looked like this. So basically, you have the origin of animals, and you have the different animal lineages branching off. So we are proud members of Balateria. These are the majority of animals you can think of out of your head. So they have two sides of symmetry. They have ectoderm, mesoderm, and endoderm. They have more or less complicated nervous systems. And their closest relatives are cnidarians, so corals, jellyfishes, and so on. Then there is an obscure group of placozoans branching here, and sponges were presumed to be the first animal lineage to appear. And this made perfect sense. So if you look at how sponges look like, they're very simple. They have about 5 cell types, one of which is this cell type where, wait, I don't have slide for this, but basically they have a cell type that has a collar and a cilium, and these are called choanocytes. Our closest unicellular relatives that are not yet animals, they look exactly like this cell type in sponge. So which made the two famous zoologists of 19th century, namely Ilya Mechnikov and Ernst Haeckel, to propose a very, very plausible hypothesis, is that once the way animals evolved is that there used to be something like this, like choanoflagellates, floating and then forming a colony, and making something like a proto-sponge. And this is actually very plausible, since some choanoflagellates do form colonies, and it looks like a sponge that got not upside down, but like inside out sponge. So of course this was a traditional view, but now what happened? Sorry, I have to mention here. So if you upset this model, this means that here the animals have emerged, and then they progressively got more complex until we reach this point that unites us with cnidarians, where we get muscles and neurons. Easy breezy. This is like the textbook example of animal evolution, except comb jellies. So once the genomes got sequenced, the comb jellies was placed as the first animal lineage, which immediately suggests something. It suggests this: since ctenophores have neurons and have muscles, there are two options. Either the last common ancestor of animals looked nothing like a proto-sponge clump of cells, but instead had neurons and muscles, or sponges and some other animals have lost all of this complexity, and then they lost all this complexity, and then it got only kept here, or ctenophores have independently evolved neurons and muscles. And this is the message that was pushed heavily by people who published the first genomes, since it's even in the name of the paper, that its implications for cell type evolution. But then the other people jumped in and they say, hey, wait a minute, you did not analyze your data right. We just take your data, we run different sequence models, different analysis, and sponges are the first ones. Then another sponges are the first ones. Then another sponges are the first ones. And obviously the ctenophore guys, they replied, yeah, but this is not, it got very messy, even like up to people citing in a paper some unconfirmed stuff. It's really, the whole field became like a First World War trench, where you have two sides, two armies, on two sides in trenches. Ctenophore guys, sporifera guys, they would sit there, they would shoot occasionally papers at each other almost every year. Sponge first, ctenophore first, sponge first, ctenophore first. And it already became a consensus that, well, we will never know what's true. Let it be. If you believe sponges, believe sponges. You believe ctenophores, believe ctenophores. And that's it. We will never know the truth. But then you remember what happened in the First World War. They invented tanks. And the tank came this year with the work of Darin Schultz, brilliant Darin Schultz and Daniel Rokshak and others, which they did not use the evolution of gene sequence, but instead they looked into the evolution of chromosomes. And they found something that I don't have time to explain in detail now, but it's orthogonal way to conclusively say that ctenophores were the first ones to appear. They found some characters that are shared by sponges with all the other animals, but not with ctenophores. So ctenophores should be the first lineage to emerge. And equipped with this, let's briefly take a look at how their neurons look like, because we can now start thinking of their neurons as independently evolved. So their neurons, they're built as in cnidarians, so there is no brain, it's just a nerve net. But their neurons, the synapses that their neurons make, they're a bit weird. So they have triad synapses that consist of some vesicles, mitochondria, and some other structures that are not or have never been reported in other animals. Furthermore, their synapses can be formed between non-neuronal cells. So for instance, neuron can form a synapse with a gland cell. So, and also it does not require exactly the same structure as synapses in other organisms. So molecularly, they're a bit weird. So knowing this, in 2018, my supervisor now built the first molecular atlas of a ctenophore, right? And he managed to find multiple cell types, epithelial cells, digestive cells, the photocytes that emit light. He even found the smooth muscle cells and striated muscle cells. So there are two muscle types. However, he could not find neurons because, so he could not find neurons, and maybe the neurons were never to be found. So this paper came out with a cell atlas of actinophore saying, here's your actinophore, here's what the cell types it contains. An obvious question is, where are the neurons? And then people later actually found neurons in this data set by looking at the expression of neuropeptides. It's just the neurons are so weird molecularly, they don't look like neurons. Because you expect a neuron in this kind of data, it's one of the easiest cell type to find. You look for ion channels, you look for synaptic proteins, you look for, I don't know, G protein coupled receptors. It's very easy in other organisms except in ctenophores. So molecularly, they don't look like other neurons in other animals. And another shocking thing came out this year. So this is a bit traveling back in history, but 120 years ago, the first Nobel Prize was awarded to two people at the same time. So this was the first time two people got the Nobel Prize. And it's even funnier because these people disagreed on a fundamental aspect of what they were studying. So first of all, the Nobel Prize was awarded to Camillo Golgi, which you may know from Golgi apparatus, and Santiago Ramon y Cajal, who was a scientist in Barcelona. So Golgi developed a method to stain neurons. Before their times, it was very hard, almost impossible to see how the neurons look like histologically. If you look at the drawings of Jan Evangelista Purkinje, you see the cell bodies, but you don't see the dendrites. So Golgi developed this silver staining. Santiago Ramon y Cajal perfected this staining, and Santiago Ramon y Cajal is very famous for his drawings of neurons. So here you have Purkinje cell, for instance. But these two people disagreed on the fundamental aspect of neurons, which is whether the neurons fuse. Well, the majority of people believed in the 19th century that the neurons, they have to form physical network. You have the neurons with dendrites, they have to fuse. And Cajal was one of, not the first person, but the second person probably, who said no. Given on my observations from the stainings, they are separate cells. And obviously they could not decide who is right in 19th century, but then in 50s, people came up with electron microscopes. They could take a look at the neurons and figured out that they're actually different cells, right? You have a synaptic cleft and they're physically separated. And this has become known as a neuronal doctrine, that neurons are individual cells, okay? And this was true until 2023, when Pavel Burkhardt in Norway discovered, by using electron microscopy and a lot of data analysis, it took them 4 years to generate a data set, where they looked at the larval ctenophore, and they found that neurons in a larval ctenophore actually form a network that's fused. The dendrites of the neurons in ctenophores fuse. They're not individual cells. So you can think of the whole nerve net as a cell. You can think of the whole nerve net in a ctenophore as a single cell. So basically, it begs the question. That since the neurons are so weird, both from their molecular makeup and from how they look like, where do they come from? And this is the question of my PhD, and I will not give you an answer because I have none. But the idea is that we will take the ctenophore, we will follow the development of a ctenophore from a gastrula stage to the adult, and we will generate single cell data sets, so cell atlases, and then we will identify neurons, and then we will try to look where these neurons come from developmentally. Because one thing is that they may be weird molecularly, they may not share any markers with other neurons, but maybe in development you still see some molecular machinery for neurogenesis, and maybe they're actually weirdos, but they come from the same evolutionary roots as neurons in mice and in sea anemone. So yeah, that's I just came back from Norway, where we actually were dissociating ctenophores. So that's Sebas and Laia dissociating the animals. So here I want to ask you if you have any questions, if we can spend 3 minutes. Okay, then we can continue probably. What? Okay. Is there some problem with the... Okay, great. So now let's switch to cephalopods. So basically, yes, I'm very glad some people are not here. So basically, if the reason why cephalopods are cool, in my opinion, is that yes, most people know that they're smart, but also the parrots are smart, right? And the dogs are smart, cats are smart, or smarter than most of the animals. What's cool about cephalopods is their phylogenetic position. They belong to this. We as vertebrates proudly belong to the group of one branch of balaterians, while cephalopods belong as mollusks belong to completely different branch of animals. And if you follow the evolution back and look at the last common ancestor between us and cephalopods, we actually assume that this animal looked like a worm or something like very, very, very primitive, which means that when we look at cephalopods, the most of things that they have about their brain is actually independently evolved. And this is what makes them cool, because in biology, in evolutionary biology, we cannot perform experiments like this. We cannot travel in time, right? So we try to search for these kind of things where the nature came up with something multiple times in order to learn some common rules. So cephalopods are a class of mollusks, and they have unique anatomy and morphology. And basically, the animals that are cephalopods are basically, the group includes octopuses, cuttlefishes, all squids, and so on. So they have really distinctive anatomy. It's very hard to miss when you see a cephalopod. And there is also a group of nautiloids that has a shell, never lost a shell, and this would be relevant for later. So first thing to mention about cephalopods is that they are actually, I mean, you may think of them who studies octopuses, but actually they have been a model for a lot of neurobiological research. So for instance, you may know that myelin is a vertebrate innovation, right? We use myelin to increase the conduction speed in the nerves, while invertebrates have never evolved myelin. So the way to speed up the conduction in the nerve is actually by making the nerve wider, right? You have higher diameter of the nerve, your electric signal conducts at faster speed. And obviously for animals like this, it's very important. The conduction speed is very important because if they want to escape, they better do it fast, really fast. So cephalopods have really big nerves that are called giant axons that have been used by Alan Hodgkin and Andrew Huxley to actually, the studies on these nerves have been used to propose a model of how the electric signal traveling around the nerves. And this is what they gained. Basically, cephalopods brought them a Nobel Prize in 1963. And the question that I will try to answer at the end of, or touch upon at the end of the lecture is basically, what are the molecular mechanisms behind the cognitive success of cephalopods and behind the complexity of their brains. And here I should put a disclaimer that for the next about 10 minutes, I will just be listing interesting facts about octopuses, because these are the animals I worked with. And if you feel lost, it's fine. If you want to recapitulate or you want to learn more, there is the whole Last Week Tonight show with John Oliver. There is one episode where he does nothing else than for 15 minutes listing awesome facts about cephalopods. So again, the next slides, they will not be connected in a logical way. It would be just fact after the fact, and then we will try to look into molecular mechanisms. Okay? So basically, first thing first, when people learn that you're working with octopuses, they say, oh, this is very cool. They have 3 hearts. Well, in zoology, yeah, you can call a heart anything. It just has to be a vessel that can pump a blood, which can be said of human heart to some extent. So basically, the animals can have multiple hearts. That's not surprising. What's really cool about cephalopods is that they have 3 hearts, 2 of which work as kidneys. So basically, you have a central heart, you have blood vessels. Sorry. So basically, you have a central heart, you have blood vessels, but then the pericardial cavity of the heart here, it's actually the place where the primary urine is formed, which means the heart is using the pressure to filter the blood through the filter membrane, just as vertebrates do in their kidneys, and this is extremely cool, in my opinion. Second thing that cephalopods are not very appraised for, and for some reason people like chameleons more, maybe because they're vertebrates. This is how their camouflaging abilities are just insane. Look at this. Look how this animal changes this, how do you call it, like the shape. It tries to mimic the coral it sits on. It's just amazing. And they can do it instantly because they have this special structure in their skin called chromatophores that can change color. A cool thing about this also is that actually, if you take out, people did some very torturing studies, very barbaric studies in 1970s, where they took out the eyes of cephalopods. And it turns out that cephalopods can perfectly adjust to the surrounding. So adjusting to surrounding, the color and the shape does not take eyes. It does not need eyes. And it's even more interesting because it seems that the eyes of cephalopods are colorblind. They cannot discriminate colors. Okay? So how do they adjust to? How do they adjust their? How can you adjust the color of yourself to the surroundings if you cannot perceive color with your eyes? Well, they can perceive colors, but with skin. So the skin contains photoreceptors that are color sensitive, and actually the skin is the side. Basically, the skin would match the surroundings regardless if the animal has eyes or not, because the skin can perceive colors. Another fact that's widely known about cephalopods, it's almost in any textbook, is that their eyes are built in a different way than ours are. So they have camera eyes as humans, but they built in much more logical way. So our high school teacher used to say that the God did not exist, but if he did, he really liked cephalopods because he built their eyes in a logical way. So you surely know that in vertebrates, retina is inverted. It makes no sense. It's completely useless, like the way it's built, not useless, but it's illogical, because basically the light has to pass through all the layers of all the neurons in order to reach receptors, and then you have to go back and put this wire somewhere, and that's the reason why we have a blind spot, completely unnecessary, and why we have like an optic nerve like this. Well, cephalopods have it built in a proper way, which means that they don't need to have a blind spot. They don't have a blind spot. They don't need to have an optic nerve. Instead, they have this region of brain that's called optic lobe, which is where the eye is directly projecting by different nerves. And this is very cool from neurobiological point of view. And actually, the surface of an optic lobe here, functionally, it's equivalent to the deep layers of our retina. This is why it's called deep retina by some people. So another thing about their cognitive abilities is the Stanford marshmallow test. So some of you may know this. It's done with kids. When you come with the kid to a room, you put them at the table, you give them marshmallow or whatever kids like to eat, and then you say, okay, I will leave you here for 10 minutes. I will leave you here for 10 minutes. I will come back, and if marshmallow is still there, you will get 2, or you will get more marshmallows. You just have to resist the temptation to eat the marshmallow while I'm not here. And the internet is full of videos. It's very funny. Some kids may pass it, some may not, and the internet is full of very funny videos with kids struggling with this test. But the idea is that this test is basically it's testing the ability of I wanted to say of an animal, of a child, to resist the temptation, right? So, and some people link it to how their frontal cortex is developed, whether you can suppress your impulses and so on. Well, cephalopods have been reported to pass a marshmallow test. In this case, this was a shrimp test. Basically, scientists taught a cuttlefish that if it waits, so the cuttlefish literally has a shrimp in front of it, but if it waits a bit, then it gets more shrimps. And so cuttlefishes are capable of understanding that if I wait a bit, I will get more food later. Another thing that may be relevant or interesting for medical people or for medical students is that is the way cephalopod brain is built. So if you think of how our body is built, we have joints, we have muscles, right? And in humans, cortex physically encodes physically encodes the contractions of different muscles. So if you get a stroke, depending on where the stroke is, you will get different parts of your body affected, right? So in cephalopods, this is impossible because the body of cephalopod is it's fluid. It has no joints. So that's why people tried, but they could not find any region of cephalopod brain that would physically encode different parts of the body in terms of movement. So instead, their solution to the problem would be to dedicate all this kind of computations to the arms. And this is known. Arms of cephalopods are extremely independent. They move as if they were different animals. And so one example of how an octopus solves the problem of bringing food to the mouse, I will give you this example. So basically, I think it's beautiful in the way it's solved because it does not require a brain. So basically, imagine that I have a food, I'm as a vertebrate, and I need to bring the food into the mouth. With a lot of simplification, you may say that the only thing that I need to do is to basically contract biceps, right? Boom. So I got the food. So I have to get the impulse from my brain for the biceps contraction. While if you are an octopus, you can touch food with any part of your arm, and somehow you need to form a joint. It's called pseudo joint, but you have to fold your arm somewhere, and it's better be in the middle between your food and your mouth. And the way octopus does it is that once it tastes something that tastes good, they taste with their arms, there is an electric impulse going to the mouth and from the mouth, and at the place where these two impulses meet, an animal makes a joint. So there is no brain involved whatsoever. And this is even more prominent in the way, like how this animal moves. Because if you look how animal moves, but different animals move, you see regularity. You look at humans, we move like left leg, right leg, leg, right leg. If you look at insects, the same. They have 6 legs, but there is always a periodicity, and neurobiologists can actually, by studying this periodicity, you can come up with very simple explanation how in brain this periodicity may be encoded. But if you look at how the octopus moves, there is no patterns whatsoever. It moves, it uses this leg, then it uses this leg, then it uses the other leg, and there is no base frequency of this movement. So this made people suppose that actually instead of having a coordinated movement in its head, the way octopus moves is that each arm is doing whatever it wants, and then the brain just says, you 7 of you shut up, and I'm following this one. And so basically, and if you look at octopus, it reminds of some kind of animal with ADHD. You know, I'm touching with my one arm, I'm touching the food, the other arm is here, the other arm is there, and then the brain just decides that I will give more freedom to one of these arms, or couple of arms. The other cool thing about them is that people have described. Wait, I will disable sound here. People have described a sleep. So basically, people that have cephalopods at home, they have been reporting for quite some time that octopuses, they have to take rest. And so this year, in 2023, there has been a study out where actually they described 2 sleep phases, just as invertebrates, where one is just a quiet sleep, where nothing is happening for the external observer, and then one phase of sleep where octopus actually moves its eyes, so reminiscent of rapid eye movement phase in invertebrates, and also you can see its change in color. So obviously this is unprovable, it's very hard to prove, but we can think that octopuses also dream. So basically, the last interesting fact, I'm sorry, there are too many, but I could not decide which ones are cooler and which ones to leave, so sorry for that. Another cool fact about them is that in contrast to vertebrates, their social life is very, very limited. So an octopus finds a mate for 2 things: to have sex or to fight. They don't form groups, they don't become friends. So one person asked me 3 days ago, how do octopuses hug? They don't hug, but you can make them hug. So there was this amazing study, really amazing study, because it's current biology. They always publish stuff like this. So basically, where the scientists gave an octopus 2 options. either play with a toy or play with a friend. That's the diagram, and it's kind of weird, the darker octopuses in cage, but in any case, there's nothing to, I did not draw this, but in any case, if you give them this option, they will go, most of the time, they will go for playing with toys. However, what if you give them MDMA? And this is what they did in the study, and this is why it's so cool. And so if you give octopuses MDMA, they actually would prefer to go to play with their friends in cage. But in any case, they would try to explore some octopus interactions over there. And this I think is just fantastic. Also, from not because it's just fun, but it also shows you some very deeply conserved role for serotonin. So again, we are talking about animals that it's all independently evolved. So basically, we are coming to the question of why cephalopods are so smart. So where does their intelligence come from? And there are multiple reasons for intelligence in general. And when we talk about vertebrates, usually people mention something that's called Machiavellian hypothesis. So the idea is that in order to be evolutionarily successful, you don't just have to be good at surviving. You have to be smarter than that guy. Or you have to know that this guy thinks about what you are thinking about that guy. So social interactions, they're actually fueling the evolution of brain. And you can see this in primates. You can see that the size of a brain correlates with the size of a group in primates. You may have monkeys that live solitary life and they have to remember one person and maybe their children. But then you have chimpanzees, you have gorillas, you have baboons, and definitely humans with very complex social interactions. And another example of this are dolphins, because, you know, for dolphin to survive, it just needs to be smarter than fish. Well, it is smarter than fish. It's ridiculously much more smarter than any fish. And one of the reasons for that is maybe that to be a dolphin, you also have to be socially very, you need to live in dolphin society to survive. So basically, in octopuses, this is not the case. They don't even have parental care. So when female lays eggs, it starts guarding them, it stops eating, and it's basically, there is a very intense physiological reaction where essentially the body of a female dies. What happens to males? They're not even there to see their children. So basically, there is no parental care. So newborn octopuses, they're born into wild without any mother teach them how to do stuff. And so the reason why this group is so extravagant and so smart is because they lost an arm race. So you know that cephalopods were very abundant. In any fossil shop, you can find ammonites, you can find these old cephalopods that are not around anymore. But then something happened in Mesozoic era, and it's called Mesozoic marine revolution. What happened is that fishes came to stage. So bony fishes, our relatives, closer relatives. And so cephalopods, they died out, most of them that had shell, and the other ones, they lost the shell, and they adopted this strategy of be smart, live fast, die young. So a normal octopus lives for about an hour, oh Jesus Christ, no, not an hour, no, a year, which is still very little, and it doesn't have shell. So basically, if you've seen this, my Octopus Teacher documentary on Netflix, it's beautiful in depicting how much danger is for an octopus out there in the wild, because anything can eat you. Even a sea star that moves 1 centimeter per hour can eat you if you don't move away. So for cephalopods, their strategy is just to become very, very smart and to avoid being eaten. And so with this, we are coming to the conclusion of the talk with basically the molecular mechanisms of what has enabled cephalopods to become smart. And the first surprise came from the sequencing of octopus genome in 2015, where they did not find whole genome duplication. Well, what is whole genome duplication at first? Well, we are vertebrates, and in our evolutionary history, there have been 2 whole genome duplications, which means that our genes and whole genome got duplicated, even twice. And some people think that we have used this in order to build our complex brains and our complex bodies and so on. Well, and that's why they assumed that octopuses did the same way. Well, this was not the case. But still, octopus genomes, or cephalopod genomes, they're very big. They're about the size of a human. And why this is interesting. It's because the normal self-respecting animal would never have such a big genome. They have genomes 10 times smaller. So basically, when the first octopus genome came out, they found that they rearranged their genomes. So there has been a lot of rearrangement of the gene order. They expanded some of the genes, and people try to link this to the function of their neurons and function of their nervous system. And here I need to mention what we did in Berlin, what we discovered in Berlin actually, because we discovered that octopuses, in addition to proteins, they've also expanded their repertoire of microRNAs. So microRNAs are this small regulatory RNAs that allow you to regulate gene expression. And so we sequenced RNA from multiple organisms. We did not, multiple tissues of an octopus, and we did not expect to find anything microRNA-wise. We only did this because nobody has ever looked into this because, well, who cares about microRNAs in cephalopods? So basically, but what we found is that here on this diagram, you see each branch is proportional to how many microRNA families the lineage has. And you see that the record is held by placental mammals and vertebrates in general. They differ from invertebrates in number of microRNA families they have, except for the octopus. So the octopus lineage, the cephalopods, are outstanding among the invertebrates. And even so, even more, if you compare octopus to another mollusks, you see that the amount of microRNAs that they gained, families of microRNAs, is even 10 times bigger. So this is a lot in terms of comparison. This is a lot given the evolutionary time. And we found out that these microRNAs, to our huge fascination, they are almost exclusively, the evolutionary novel ones, they're almost exclusively used in brain, and they're probably functional. And so we assume that these microRNAs were at least in part what allowed cephalopods to build their complex brains by encoding more different types of neurons. Well, you may ask, okay, how many neurons does an octopus have? And we don't have an answer for this. So here I have to mention a great study that I also had the pleasure to co-author, which was led by Ruth from Yves Sauntien's lab in Belgium. So what she did, she took newborn octopuses. These are the only ones that EU laws allow you to work with to not violate animal welfare. She took their brains. They have small brains, but we assume they already harbor the majority of diversity, and she did single-cell RNA sequencing on them. And what she found, she basically found dozens of different neurons. She found glia. She found even the neuronal types that have different neurotransmitters in the same cells. So you can have dopamine and glutamate or something of this sort. And this is very exciting. This is what they state in the paper, is that, yes, this is high diversity, but a valid point is, well, how does it differ from other organisms? And the honest answer is we don't know. We don't know because we don't have data. We don't know how a normal mollusk's brain looks like in terms of different cell types. So what would be a good reference for this? And this is my offer, not my offer, but if I had unlimited amount of time and money, what I would go is that I would go for sequencing nautiluses. Because these animals, remember, we talked about cephalopods losing their shell, trying to become smart and so on. That's all true except for nautiluses. So for God knows what reason, they survived, and they're a real evolutionary fossil. They have never lost their shell. They don't have camera eyes. They don't have complex brain. They just look very similar to how the last common ancestor of cephalopods looked like. So in order to learn about these brains, we actually need to go and study nautiluses. And I would love to do this, but they're not allowed to be. It's very hard to work with them. They live very deep, and they live for 20 years or so. And then the only place they have them at in Europe, I think, is Monaco. In any case, this would be a great model system to study further. And with this, I wanted to summarize that, yeah, cephalopods, they have unique anatomy and morphology. They have complex behavior with all these weird facts that we touched upon. They have multiple genomic mechanisms. And of course, I'm biased to say that these are microRNAs because we got involved into this. And there is still a debate what makes cephalopods smart, whether it's just the number of neurons, just the number of neurons as in dogs, the comparable number of neurons as in dogs, or maybe they have some novelties, novel cell type, novel neurons that make these brains more more efficient somehow compared to other mollusks. And with this, I want to thank basically all these people. There are a lot of people involved in this. So people in Berlin, my supervisor Nikolaus for paying me, and also my father for paying while I was in Berlin. My current supervisor Arnau, Xavi, Sebas, Laia for working on Ctenophore project. Also Norwegians, and also Ruth, who was working with me on Octopus, and now she moved very conveniently to Bergen to study Ctenophore. And then Bastian and Kevin, these are the persons who annotated microRNAs, and then, yeah, people who decided for some reason to pay for my PhD, and our armed forces of Ukraine for defending my home, my family, and you for being an amazing audience. Thank you.